Good afternoon. I'm very happy that many of you stayed. I know we had some very stimulating talks earlier, and I was worried if you would get tired and leave. So I'm Dr. Ashton Rogers from the University of the West Indies. I'm located in Trinidad and Tobago, all the way down at the end of the Caribbean. I noticed that many of you presented pictures of your place of interest and where you work and what it was known for. So I'm from Trinidad, and it's known for nice beaches. So I'm introducing my place of work. Some of my ideas actually came looking at that view. So let's get down to business. I'm really excited to discuss my research on the effect of systemic inflammation induced by a high sucrose diet on inflammation in the rat substantia nigra. I'll go through some background to give you an idea of how I arrived at this concept and this line of studies. So Parkinson's disease is the second most common neurodegenerative disease worldwide. We recognize this. But in my opinion, I think it's possibly the worst age-related disease, one that we fear a lot, because it not only incapacitates physical movement and making the smooth body movements rigid, causing tremors and disrupting your natural life, but it also slowly incapacitates the mind. It could cause depression, dementia, cognitive function. So it is really a devastating disease. Although we have learned a tremendous amount about Parkinson's disease through all the exciting research that we've learned some here, and of course we'll review online, 80% of Parkinson's disease is still idiopathic. So we're making strides, but 80%, that's alarming. Now, many of us are familiar with the information on this slide, but just to review and recap, we know that genetic mutations and aging can predispose healthy neurons to becoming injured, and of course that triggers the activation of microglial cells. They could remain primed for some time where you have no actual pathophysiology in the brain, or they could be pushed into becoming activated, and then we have the phagocytic versus the non-phagocytic, which is associated with all the pro-inflammatory cytokines and neurotoxic reactive oxygen species. So this story has been told many times. We've recently come to realize, and we were just talking about it, that systemic inflammation through infections or toxic chemicals, as in animal models, can drive neuroinflammation. There are many studies that show that if you superimpose systemic inflammation on an existing neuroinflammatory process in the substantia nigra, it makes it worse. So this is of importance, right? But when we look at the global distribution of Parkinson's disease, we see that it affects people across the entire globe. So the problem with this model that we have established so far to understand and explain Parkinson's disease, if we use genetic mutations, we wouldn't be able to explain the worldwide distribution. We could see, hereditary traits here and there, and we might see patches of Parkinson's disease. And if we use infections, again, places with a high number of communicable diseases, you would see probably a high level of Parkinson's disease. So you could kind of associate it. But what we see from data on the global distribution, it's widely distributed. So even though we know this, in reality, it doesn't really account for much of the 80% that's idiopathic. So what I'm proposing today is metabolic syndrome as a possible contributor to all of this here. Metabolic syndrome by itself obviously doesn't cause Parkinson's disease. I mean, we would have seen that coming a long time ago. But what I'm suggesting is that Parkinson's disease may be triggered by genetic mutations or even infections, right? The thing with infections is that it creates an acute inflammation. But Parkinson's seems to be driven by systemic inflammation or any sort of inflammation that's sustained, chronic inflammation. So that's probably the reason why we're not seeing a direct overlap with infections and Parkinson's disease. Although some places, there are reports showing a correlation with, for example, like influenza and stuff and Parkinson's disease. But I want to review this new disease that's taken over the world, and that's metabolic syndrome. Now, what do we know about metabolic syndrome? Well, metabolic syndrome we know is a cluster of metabolic anomalies that has to do with obesity and type 2 diabetes. And with respect to obesity, I'll just key in into the main points and leave out some of the details. We know that in an obese person, we have hypertrophic adipocytes. These adipocytes are hypertrophic because they are lipid-laden. That tips the cells into being in a sort of an infected state. So they begin to start to produce pro-inflammatory cytokines like TNF-alpha and so forth. And this causes the infiltration of monocytes, which differentiate into macrophages. And you can see that in the adipocyte of an obese person, there are elevated levels of pro-inflammatory cytokines. What is also known is that this leaks out into circulation, and it can travel to the liver, and because the liver has matching cells, the Kupffer cells, the same cells that would move into the adipocytes and cause inflammation. Similarly, you have non-alcoholic fatty liver disease. So now we have, in an obese person, there's a possibility that you could have systemic inflammation produced by the adipose tissue and by the liver. And these cytokines can now leak into circulation. We see from animal models that systemic inflammation could actually exacerbate neuroinflammation. So what about the effect of systemic inflammation as associated with metabolic syndrome and neuroinflammation? There's limited information to explain this. By looking at the literature over the years, I just took a few to spot check. What we see with obesity, several basic parameters we use to try to find a link between obesity and Parkinson's disease. So researchers have been taking a glance at it from time to time over the years. And what we see, they've used either body mass index, or waist circumference, simply being overweight in several studies, but we have mixed results. Some results, some show no correlation and some show a correlation. But it does tell us that there's some concern. There's some literature that also support that diabetes by itself might actually affect the vessels in the substantia nigra and cause some sort of oxidative problem and might trigger Parkinson's disease. But this, obviously, you could see there were just a few studies. These were the two more detailed studies, and they show mixed results. You know, one supports, one doesn't support. So at the moment, there's no clear literature that shows or strongly supports, you know, a correlation between metabolic syndrome, obesity, or type 2 diabetes. However, though, when I look at the statistics from WHO, CDCs around the world, UNESCO, and NIH, this graph on the left shows mortality related to Parkinson's disease. And of course, the first glance, you could see that from high to low, it's distributed across the world. When you look at the graph on the right-hand side, you can see that there's a similar distribution with obesity. But what is even more alarming is what I've circled in red. In the developed countries, where non-communicable diseases have replaced communicable diseases, so low infections, we see what? High amount of obesity with an overlap of a high mortality related to Parkinson's disease. So, possible there is some sort of connection. So this data has had me going for some time to try and figure out why aren't we looking at it, or why aren't we really wondering if it could potentiate. So the premise I came up with is that if indeed the aging and genetic factors and even infections could prime the microglial cells in the brain or activate it or something and get it going, but it stays in a benign state. So not everybody develops Parkinson's disease, right? However, though, if you have the superimposition of the systemic inflammation associated with metabolic syndrome, maybe it could tip the scale. Maybe that's what's driving it, because clearly what we should have seen here is like Africa with infectious diseases should have been red, but it's not red. That's a place that has a lot of infectious diseases. So clearly the data is making us think that metabolic syndrome might have a part to play. So based on the review data, I came up with this hypothesis that systemic inflammation induced by a high sucrose diet can exacerbate an ongoing neuroinflammatory process in the rat substantia nigra. To find some basis now for future either clinical studies or more animal study research. So in order to investigate this, I use one of the classic models for introducing an inflammatory process in the substantia nigra, the model established by Castano et al. The rats were sedated. They were then placed in a stereotaxic apparatus using the coordinates. I then drilled a hole here and do a substantia nigral injection of 2 micrograms of LPS lipopolysaccharide, which I think most of you know. So this is a classic model. And then, of course, the rats were sutured and allowed to recover for 1 day. Preliminary studies show that I was able to target the substantia nigra. So this shows drug delivery, and this shows that the LPS was actually causing neurodegeneration. There are more immunocytochemical analysis, but I'll save you the trouble of all of that, just for you to get the idea of what I've been trying to accomplish. So I have a working model here to introduce neuroinflammation, and there are other slides. I looked at microglial cells, I looked at gliosis and stuff that support the typical model. In order to investigate the effects of metabolic syndrome, prior to this work, I did a lot of research on different diets. And even this model too, I was a bit skeptical about using it because initially I wasn't convinced that it was doing something. But that was a good thing. That actually reinforced what we know from population and human studies. Now, like I said earlier, people with metabolic syndrome do not naturally develop Parkinson's disease, right? So when I first used this model, I went straight into the brain and did immunocytochemical and PCR analysis and didn't find inflammation in the brain. So it did actually support what human studies show, right? Now, what I did, a very simple dietary regime, I used 30% sucrose in the drinking water. And that's what I fed the control animals. The experimental animals. The control animals simply received normal drinking water. And all groups received the same standard rat chow. So the only thing that was changed was the sugar in the water. I sort of reciprocated what we do with soft drink. Coca-Cola is 30% sucrose, or probably even more. Most sugar-sweetened beverages is about 30%, 30 packets of sugar in one serving. So, of course, we could debate about the metabolism rate of the rat and stuff, but from the results I got, I was very impressed and excited to tell you about it. So, when I fed the rats this for 30 days, as you can see, they developed dyslipidemia, they had hypertriglyceridemia, they had increase in total cholesterol. The rats had excessive adiposity when I was actually doing the perfusion for, I just noted this. It wasn't a variable that was measured, it was just an observation. What I did find though, is that even though the rats had these variables, these changes, the weights were the same. So that was a bit confusing analyzing the data initially. The rats maintained the same weight, so both the sucrose fed and the controls, but the body composition was very different. If you picked up a rat that was fed sucrose, you could tell that it feels lighter. The lean mass changed, okay? Now, when I looked at, of course, in order to satisfy the model of metabolic syndrome, they must have changes related to obesity and changes related to glucose metabolism. And they displayed a small degree of impaired glucose tolerance using a glucose tolerance test. So now what I did was simply superimpose the two models and see what the outcome was. In order to analyze, I did two sets of analysis. I wanted to see what was going on with the morphological changes in the brain, and I also wanted to see what was going on with the markers for inflammation. So I did PCR and I did immunocytochemistry. I mean, this was fun. And of course, reading all the articles by folks, I just love immunocytochemistry. So what I did, I used anti-TH to mark the TH positive cells. I used anti-GFAP to monitor gliosis, because the astrocytes can also contribute to inflammation in the brain. Of course, I looked at, I used the OX6 monoclonal antibody to target the activated microglial cells. And because I was actually injecting in the brain, it was breaking the blood-brain barrier. So I looked at the degree of inflammation of the monocytes from the blood, because these three will all contribute to the degeneration through neuroinflammation. And with respect to PCR, I looked at an array of cytokines. I just wanted to be sure about what I was, about the conclusion I was going to make. So I looked at all these pro-inflammatory cytokines, some protective anti-inflammatory cytokines, some macrophage markers. I also used PCR to back up the changes. So I looked at the mRNA expression of TH and GFAP in the substantia nigra. And of course, I wanted to confirm that there was systemic inflammation. So besides using the typical markers of obesity like triglyceride and HDL and total cholesterol, I also backed it up with some markers that would identify if the adipocytes in the sucrose-fed animals were actually pro-inflammatory. And I not only looked at these in the adipose tissue, but I decided to look at it in the liver, because the premise is both the adipose tissue and the liver contribute to the systemic inflammation. So data from this give me an idea of the systemic inflammation, and data from here give me an idea of the neuroinflammation. I basically had 5 groups. One group was injected with saline, so that was like baseline for all the statistical analysis. Of course, I reproduced the actual model that was established by Castano et al. Using two microglia, using 2 micrograms of the actual drug, LPS, and I injected this in 2 groups. One was given normal diet, and the other one was given the sucrose. And what I did some preliminary studies to check a dose effect on this drug. And I simply kept diluting the drug until I got a concentration that would produce barely any inflammation in the brain. So what I wanted to do was to find a model that would represent a benign inflammation. So this is a person probably who is now developing Parkinson's disease. I hope you get that idea. So I wanted to see what it would do with the very initial stages of neuroinflammation and, of course, a more chronic inflammatory process. So with this low dose, it actually ended up being a 1 in 10 dilution of the normal dose. So basically, I injected 2 micrograms and 0.2 micrograms diluted in saline into the substantia nigra. So I basically compared these data from the 4 groups. Of course, everything was analyzed using statistical programs and over-testing and stuff. So I'll first just take you a walk through what happened with the immunocytochemical analysis, and then I'll tell you about the PCR analysis. What do we see here? Well, this is simply reciprocating what would you expect when you inject 2 micrograms of LPS into the substantia nigra. So you can see that there are some activated microglia, there's some gliosis, and there's some infiltrated monocytes. So there is some degree of neuroinflammation, and I can see that there's a certain degree of loss of the Th-positive neurons. Now, what I did to analyze this properly, I used ImageJ and did densitometric analysis on the slides. So took pictures, analyzed them properly using our software. So it's not just visual, this is just to help you understand. So everything was statistically analyzed. Now, look at what happens when I actually match up the data from the same group that was injected with 2 micrograms, but fed sucrose. And, well, this is not a good representation, but when I zoom out, you actually lose in the quality. But what I saw with respect to gliosis, it was widespread. What I saw with respect to the activated microglia, it was also widespread, and there was a higher density. I didn't intend to do cell counting, but I needed more equipment to do it. And you could see that the site of injection along the needle track, there were more AD1 positive infiltrated monocytes or macrophages. So roughly, from the densitometric analysis, you could see that there was an increase in the loss of Th positive cells and the cells that actually elicit neuroinflammation in the brain. So this was interesting because it was only 30 days. So this got me going. Now, what I want you to appreciate here is the data from the low dose. So what I was able to do is to create a very low degree of gliosis, but look here, this is the most important slide that I want you to pay attention to. With this dose, in 30 days, the lesion healed itself, in summary. There was no activated microglial cell, meaning that the inflammation actually dropped down to extremely low level. So with this dose, naturally, it would heal itself, right? So it's like an infection. It will go away, or it's like trauma, it will heal itself. And there were just a few infiltrating monocytes. So this contributed to a very low level of inflammation. So there was still something very, very low. I'll show you the graphs from the PCR data after this. But look at what happened with sucrose. Sucrose actually potentiated and exacerbated the neuroinflammatory process in the brain by significant folds. You can see here widespread distribution. If I zoom out, well, you'll see how wide it was almost throughout the substantia nigra. It wasn't like this. This is what I expected, and what I found was totally unexpected. There was also widespread gliosis and widespread loss of Th-positive neurons in the substantia nigra. So this was shocking. It took me a while to understand. I thought I did something wrong. How could the low dose actually have a worse effect? Usually that doesn't happen, right? That's atypical. If you increase the dose, you get a greater effect. But the substantia nigra does not behave like that. Apparently, because if you, let's say in some parts of the brain you have trauma, the brain will heal itself. But when the inflammation gets going in the substantia nigra, it could take different parts, because we see only parts of the population actually having Parkinson's disease, even though it's a significant amount. Now, just to wrap up the story, this is the normal dose and water, and this is the low dose and sucrose. The degree of inflammation here should not be higher than the degree of inflammation with the higher dose, but it was. Just to support this data with the PCR results, I looked at the markers for the macrophages, and just to make it easy and quick, I'll simply focus on the group in purple, which is the low-dose LL, low-dose LPS, and sucrose. And when I compare the data and the multiple analysis, I left that out just to make it simple. So what you're looking here is just data compared to the saline injected group. But in this group, you could see that it had the highest mRNA expression of the macrophagic markers. You could see that it had the highest levels of the pro-inflammatory cytokines. I also found that the protective anti-inflammatory cytokines, also there are some reports that said the anti are also increased in neuroinflammation. So this was a bit confusing for me. I'm still trying to investigate an explanation for this, but this is the data that I found, that they were actually low. So I guess I could go with the articles that said that high levels of anti-inflammatory cytokines protect. And since this group had the worst, well, then maybe they weren't being protected well. So I'm still trying to go through the literature to understand this. And then the GDNF and BDNF are supposed to be neuroprotective, but they were low in this group. So it seems as if this group, once the inflammation got going, the protective factors reduced for some reason. I'm still this slide, I'm trying to get the explanation for it, and that's a limiting factor in publishing this paper right now, but I'm searching for the answers. Just to present you with some data from what I found from the systemic inflammation. So this shows you the adipose tissue and the different inflammatory markers that I looked at. So this is to get an understanding of if they had infiltrating macrophages from the blood, or if the Kupffer cells were actually producing inflammatory cytokines. And what I found, again, in both sucrose groups, they had elevated levels of the macrophagic markers, the pro-inflammatory markers. So it helps me to appreciate the fact that, yes, there was some sort of systemic inflammation that's taking place in the adipose tissue and in the liver. Well, I'll show you the data for the liver now. So I found similar results in the liver. So there was cytokines, pro-inflammatory cytokines coming out of the liver, coming out of the adipose tissue. I could have looked at more markers, but I wanted to get a picture of what was happening. So based on this data, I'd just like to conclude the novel findings. So I was able to show that there's a possible correlation with a 30-day sucrose-enriched diet that introduced features of metabolic syndrome. So both obesity markers were changed, and the rats had impaired glucose tolerance. And of course, they had pro-inflammatory cytokines being produced by the liver and the adipose tissue, so they had systemic inflammation. And I found that this can exacerbate an ongoing but benign neuroinflammatory process to a pathological state. That's how I described it, because it seemed excessive, more than what a standard dose would do in the substantia nigra of the rat brain. So what does this actually tell us? This research actually gives the foundation for future studies that would look at the relationship not only with systemic inflammation associated with infections and toxic chemicals like pollutants and herbicides, pesticides, rotenone, and all those other things, but it would, I'm hoping to bring systemic inflammation through obesity, through metabolic syndrome, which is what most of the developed countries are suffering from now, to the forefront of research, along with all the other fascinating stuff that have been discovered. I would also want to mention that since the rats that I looked at did not have a change in weight, using things like BMI and overweight might not be a good criteria for trying to find a link between obesity or metabolic syndrome and Parkinson's disease. So maybe we have to review the markers they're using to identify metabolic syndrome or obesity to correlate it with Parkinson's disease. And I think that it would be more important to actually measure the level of systemic inflammation and try to correlate that in a human population, which is easy to measure, and map the patients until they develop Parkinson's disease, or see how their Parkinson's disease develop. Now, the interesting thing about Parkinson's disease is that when motor dysfunction begins, the patient starts to lose weight. And this effect in some patients actually would probably mask the obesity thing, because when we look at some Parkinson's disease patients, they're not overweight. So it probably gets the process going, but when all the clinical signs and symptoms of PD onset, the patient now starts to lose weight. Well, some of them actually. And to talk about the multivariate component of Parkinson's disease, it may be as a result of genetic, aging, probably some systemic inflammation through infection, primary microglia, and maybe the systemic inflammation due to being overweight just pushed it over the edge. That's a new concept, I suppose, or a probability. And I think that we should start looking in that direction. So thank you very much for your time. I hope you appreciate